So I'm happy to moderate this block 2, which is entitled Translational Toolbox and Pipeline. Block 2 is the same length as block 1 in time, but it has twice as many speakers. That means each speaker gets 10 minutes. That also means I will not be able to give you deep introductions. And it also means I'm going to rely on you basic and translational scientists to show discipline in time control. So without further ado, a man who doesn't really need introduction, Joe BUXBAUM.
So I'd like Mustafa to come and moderate my talk. Alex agreed to go a little shorter, so I was able to take some of his time. Anyways, if I just hit forward, yeah, that's me. So translational toolbox, we'll go pretty quickly. I only have 140 slides, so we got 10 minutes. So basically, when you think about the translational toolbox, it's about getting through this pipeline. It's about getting through this pipeline of, identifying a high for a genetic disorder. The first column over there, thank you, a high-risk gene, okay? Convince yourself that it's a target. You might want to make human neuronal models, right? And this is the drug discovery part for small molecules. So you might use these cells for screening, and then you look at your genetically modified animals, and then you go to patients, and you even start with patients, first of all, to generate some of these tools, but also to do all the phenotyping you've heard about, right? So we have endpoints, and we have biomarkers, and so on. And the new thing that's just really emerged in the past couple of years as a real alternative to small molecule screening is, of course, gene therapy. Some of the work with in spinal muscular atrophy and other disorders like that have made us realize it's something we can actually do now, and you'll hear more about that. So the only difference is that instead of screening your neurons for small molecules, you develop gene-targeted therapies and you apply them to your various model systems, right? So that's the translational pipeline writ large. And I'm proud to say that the Seaver Center does a lot of work in this space, and our main gene historically has been SHENG3, and we try, as you've heard, Alex already talked about seeing families and patients, having model systems, which I'll talk about briefly, and trying to go back and forth all the time. And we're not the only ones, of course. So a little reference to pop culture and to some old poems. I'm supposed to talk about genes, cells, and animals. So this is, Flo Rida, Poe, and Robert Burns. If you can't see it, okay. So, it's been a long road. I mean, it's been a very long but fulfilling road. I don't know how to say it. This was in 2010. Where's Paulina? Paulina was adamant that we have a symposium at IMPAR in 2010. She's gone, she's left for the day. But this was our first presentation. We made a mini symposium that was at the same time at IMPAR, or INSAR now, and it was on at CHOP. And we presented some of our first data. We had other scientists speaking as well. This result, I always come back to, that in a heterozygous animal, the dynamic synapse changes are not stabilized. And we actually tried IGF-1 and presented it there, including a prior neuron compound, the first three, the tripeptide, which was presented there as well. So, genes. And I think this is my subtext right here, which is that biology is infinitely complex, and that's kind of why we do it. If you remember when you read Zen and the Art of Motorcycle Maintenance, Pirsig almost lost his mind because he found that every experiment didn't answer a question so much as it generated additional questions. And if you love that, you're fine. If it drives you crazy, you probably shouldn't be in that business. But the beauty of working about a clinical disorder is that we don't have to embrace all of that complexity. We can use our tools to say, this is interesting but not relevant for drug discovery and development. And I do feel, you know, there is a schism between kind of the basic scientists like myself and families that are very kind of focused to make sure that we're actually talking the same language and realizing this is super interesting, but it may not change anything for my child, right? So I think that's, yeah, it happens all the time. So so I think the most important thing is always to go back to the genetics. Tess did a great job summarizing this. I would say that we knew about PMS since the 70s because of Ring 22, which he already talked quite a bit about. All the phenotypes that we see in PMS were described systematically for Ring 22 50 years ago. And then more and more work by great clinical geneticists to find the region and then the gene. I won't go into it. And then this paper, of course, where the first point mutation was identified in individuals with autism. And now if you go to large-scale databases, both with disorders and without, we know a lot of variants in the gene. This is ClinVar down here, so gene mutations across the gene produce a phenotype. And conversely, and this is really important, if you look at controls, defined controls, you don't see this, right? So again, there's always something on the margins of science that maybe somebody has an inherited variant, maybe there's some more common variant that contributes risk, but we're 99% sure, which is good enough to go forward, that the people, typically developing individuals, do not have a deleterious mutation in this gene. We have data on hundreds of thousands of individuals to support that. A scientist will tell you, what about a missense variant that's inherited? Sure. But again, from the point of view of drug discovery and development, we can go with the 99%. It's kind of like global warming, right? There's some people who say it's not happening, but most people agree. So it's a question of our level of confidence. And we're very confident about SHENG3 being de novo mutations driving the phenotype. And then you've heard also, once we find these things, we and others spend a lot of time doing in-person phenotyping. This is Latha Surya. So she just was on the panel. This was the deletions. And when the point mutations began to come out, we felt very strongly that we had to study the point mutations and make a comparison. This was Sylvia was doing this together with Alex and Paige. And then you've already heard from Tess that as the RDCRN that Mustafa put together was created, we could do this on a much larger scale across multiple sites, right? And fundamentally, you know, to a first approximation, the phenotypes are similar, whether there's a deletion or a point mutation. There are some differences we can talk about, but we're pretty sure, I think we're at the 95, 99% confidence that SHENG3 is the major driver of is sufficient for the phenotype. And you can keep on going back to your clinical samples. This is looking at blood and showing some differences. As the deletion gets bigger, some of the cells in the blood actually change. And we're pretty clear about that now. So you can find differences phenotypically to some degree, but neurobehaviorally to some degree, but even in blood markers, right? And the other thing that we know is that this is a dosage-sensitive gene. So 0 copies is not found in the clinic. It's an interesting biological model. 1 copy is what you find in the clinic. And it turns out that 3 copies is deleterious as well. So triplication of this region, which actually isn't as rare as we think, produces a phenotype, and it tells us that like most dosage-sensitive genes, it's a U-shaped curve. So you have to kind of think about that in the therapeutic domain. All right, so what about cells? There's a massive literature long before iPSCs of really good neuroscientists working on cells. You gotta be kidding. Anyways, and it really is painful to go through this literature because it's so vast, but it's very important. This is a paper from 2005 that shows that if you take a neuron that should not have a synapse, a spine, and you put in SHENG3, you actually create the spine. So when we think about putting SHENG3 back in the brain, we have to ask ourselves, are we going to change the properties of neurons that shouldn't have had it in the first place, right? It's a non-trivial question. The other thing about this, and this is an important one too, again, for the point of view of what we think we need to know, is whatever you see by knocking down the gene was reversed by putting back the gene. So there's really interesting questions about, you've heard about last night, about fusion proteins, fusion products between SHANK3 and ACR, for example. But in the in vitro space, we can just put back in just SHANK3 as a cDNA, and if it recovers the function, we can go ahead from the point of view of clinical trials, even if it's still a fascinating question about that fusion protein. Here's another example. We put SHANK3 back in, but it's a little more complicated here. You get a reversion of phenotype. Here's a study that looking at an ASO at the downstream region of SHANK3 showing upregulation. You've heard about clinical trials going on with this. And we've published a paper on a handful of cases, and we've talked to Kate about kind of cataloging those and letting everybody know that they're available if you want them. So these are iPSCs, patient-derived. And at the PMSF meeting, the CBER Center is going to go there and collect bloods and then bank them and store them as frozen cryopreserved cells so they can be reprogrammed in the future. Another kind of cellular study that one can do is, of course, heterologous cells. This is the work we're doing with the Disney lab, where we're using the very stable 3' UTR of SHANK3 that has a very characteristic secondary structure, and we're using it to screen for drugs that upregulate the expression, and we're using a collection of compounds that have been kind of chosen to be drug-like and target RNAs. And so there are 2,500 compounds in the library. We had 16 in the first pass. We did some further screening, and then ultimately there are 5 that survived that show nice dose response curves. Here are 3 of them, and anybody who knows a good cell line, because we're lazy, that expresses SHANK3 that we could throw this on, we'd be happy to do it, because we need to look at endogenous protein and then SAO, structure-activity relationships, and then take it from there. NIMH does have iPSCs. Some people are reluctant to take cells from the NIMH repository because it's not always clear what the technology was to reprogram them, the quality of them. It is a suggestion that the foundation could support getting those cells, having them QC'd, and make a database, because everybody can access them that way. Mice and monkeys, I'll be quick. There are lots of knockout mice. This is a table from a few years ago that we kind of put together, everybody's knockout mouse. There are some pretty strong similarities, in terms of motor behaviors, social deficits, or repetitive behaviors. There are differences, certainly, and it depends on labs and strains and all that. Part of the difference probably maps to this slide from Yonghui Zhang's lab that he showed that there are multiple start sites and there are multiple products. And so he and we actually made what we call a full gene knockout, for better or for worse, which is from exon 4 to 22, and it has interesting properties. It's a more severe phenotype by a bit. You've heard about Hala's work on the rat model, so there's many mouse models, and there is a rat model or 2. And again, you could do very elegant experiments. This is GUOPING FENG study, taking SHANK3 and turning it back on at different stages of development, and then showing recovery, right? So, critical windows. You could do many things, and you could ask very specific questions in these models with their limitations. Alex talked about moving from mouse models to clinical trials with IGF-1 and then to growth hormone. And this is work that Pauline helped stimulate, which is looking at another compound in the mouse model. But what we haven't done, the mouse models are not perfect, right? We know some things, and you've heard about them in Phelan-McDermid syndrome that are not captured well in the mice. And this is the family from the 2007 paper, and you can see they had clear regression, unquestionable regression, including late life epilepsy that wasn't there until they were 19, right? And both of these boys passed away, right? So again, if you look carefully, this is the supplement of the paper. There's a lot of evidence in the literature about regression, about many of the phenotypes. So epilepsy and regression, we are not modeling well in the rodents. And that's a question. I mean, Mustafa's done some work at his consortium looking at some changes, epileptic form, some interictal spiking. Is that what that is? Am I confused? Yeah. And so there's a desire to push that forward. There are meetings about this. And of course, April's going to show this slide again, I'm pretty sure. But, you know, the question is, can we take some of the things that are not clinical, like frank epilepsy, seizure disorder in patients, and model something different, like perhaps this phased amplitude coupling shift that you've heard about from Alex. April, I can take your time now because we've given your talk. And then, of course, Xiaoping Feng and one of his lead postdocs, now independent faculty, have developed monkey models, which you hear a lot about. And in terms of availability, at least of the mice, JAX has many of them, and actually it's kind of a who's who. GFN is Xiaoping Feng, Yonghui Jiang, I don't know who Bux is, you know. So the people in this room have made sure to make their animals available. So I think I can stop there. Apparently, I'm stopping there.
Well, hello, everybody. I'm thrilled to be here. My name is Anna. I'm a neuroscientist working with CureSHANK to help them convert science into a pipeline. And I'm also a drug hunter, so let me tell you how I look at Phelan-McDermid from the perspective of a drug hunter. The first thing that stands out to me is like, this is a large, rare disease. We heard before 1 in 30,000. We also have estimates for 1 in 15,000. So this is the size of several rare diseases that have multiple treatments approved, that have late-stage clinical trials, and we're not there yet. The second thing I noticed is that this disease seems to be in the hotspot for many of the companies. So from the biological perspective, it's a haploinsufficiency. A lot of technology companies are interested in that. And then from the disease area perspective, this is autism, neurodevelopment, there is no shortage of industry interest in the space, but we don't have the pipeline yet. And the third is that I think you will believe me this because we've heard a lot about this today. There's been a substantial amount of work done by this community to de-risk the preclinical and clinical space, very good preclinical models, reversible, a lot of work on outcome measures. I was impressed with the natural history efforts. And a very large and engaged patient community. So trials should be feasible. And we're not there yet. And that is the goal of CureSHANK, to cross that bridge and turn that science into a pipeline so we can have those treatments. And was also the plea of the moms this morning, right? So let me tell you about the pipeline we could have, not the one we have, based on the biology of this disease. Because what it has is that it gives us many entry points as drug hunters. For example, you could think of developing a gene therapy, straightforward gene therapy, gene replacement, and that's what Jaguar just got an IND to do. But because it's a haploinsufficiency, you could also think of doing this other gene therapies that act on the good copy to boost it. This can be zinc fingers, dead Cas, there are many options. Other companies might want to use some gene or mRNA editing for a subset of patients that might be amenable to that. But I expect more companies to be interested in, hey, this is a haploinsufficiency. Let's boost the good expression with some ASO. And there are some early programs in the space. You could also think of trying to compensate for the protein or synaptic problems, or most likely develop a symptomatic treatment that could partially also account for the synaptopathy, and that's what Neuron just showed in a phase 2. So we could have many things. And the good news is that we start already having an early stage promising pipeline. And this is big because if everything goes well, by the end of this year or next year, we're going to have our first pivotal clinical trial and the first gene therapy. Like, this is big. A lot of rare diseases don't have this. So that's the good news. The bad and the ugly is like, this is not enough. We know that drug discovery and development is a funnel, and many programs have to come in for a few of them to come at the other side, so we need to have more. And I wanted to use these few minutes to frame that. So today, you are our brain trust. And what we want to hear from you in this blog, that is the translational, is, do we have enough science to build a pipeline? And I just need enough, not perfect. We were saying before some questions are not needed, so do we have enough of the needed ones? And in the next sessions, how can we feed that funnel by converting more companies? And how can we make sure that more of them are successful so that we can have all those treatments? Thank you.
Guoping, you're up next. Talk more about the basic biology.
So I was asked to give a 10 minutes talk about the introduction about the basic function of SHENG3. The function of SHENG3 is very, very, very complicated. That's all. You want to know more? I can tell you more. Seriously. So we have 80 billion neurons in human brain, about 86 billion, and probably 100 billion of astrocytes. Neurons, unlike other cells, they usually don't function autonomously. They have to connect with other neurons, and whatever you do, there are millions of neurons working, whatever little thing you think you are doing. So they communicate mostly through synapses. So SHENK, including SHENK3, is the key protein to help build and build the synapse and help function and plasticity. So SHENK is one of the proteins at the synapse called scaffolding proteins. So Morgan Shen, who is originally discovered the SHENK family of proteins, at least one of the groups, and so scientifically he's the grandfather of SHENK. And so there are 3 SHENKs, SHENK 1, 2, and 3. They are distinct genes, but sequence-wise, protein structure-wise, they are very similar. And there are some differences. The major difference we found is that many others also found is that expression patterns. There are mutations found in patients for SHENK 1, SHENK 2, and SHENK 3. SHENK 3 is the most severe one caused symptoms. And we think one of the reasons is probably because functionally, structurally, they are very similar, although no one has done the experiment, put SHENK 1 or SHENK 2 into SHENK 3 mice. Do you rescue them? No one has done. But based on structure analysis, they are very similar. Probably they will rescue most of the problems. But the expression pattern is they are actually in the striatum is only SHENK 3. And even you delete the SHENK 3, SHENK 1 and 2 cannot be upregulated, compensated. So that's done in mice. So because these circuits, the striatum is a major part of the corticostriatal-thalamus cortical circuits, which is dysfunction has been linked to many problems you can identify in the SHENK3 mutation patient. So including habit formations, cognitive functions, and social interactions. So, and you all see these papers before. And as a neurobiologist, when you have gene, we thought, we got it. Now you have a gene, we can make models, we can figure out everything. It's not a big deal. So 3 labs, especially Joe BUXBAUM lab, published the first paper on the mouse model. Yonghui's lab and my lab, we also generated a mouse model, and we thought, we got it. And so we had the best idea in the world. We're going to study the mouse model, find a converging molecular target, we're going to cure it. So very simple. 20 years later, we still haven't done that yet. So, but it's clear, it's critical for the synapse function. I'm just show a few slides from my lab, but many other labs have done just for simplicity. I'm just use some of the data we generated. So it's really important for not only for the function of synapse, but for the structure and development of synapse and for at the molecular site. So scaffolding protein, which is form a scaffold, can bring many other proteins to the synapse. Not only bring them once or twice, it's dynamically based on the need, and based on different cell types and different development times. What do you need? So it's a really wonderful protein. It's just very difficult to fix it when you have this kind of multiple functions. And just like in your company, the person can do everything, then she suddenly left, and now you're in trouble because you have to find 10 different people to replace this one person. So that's almost like this kind of thing. So, but the good thing is that with the mouse model, we see a lot of, we call the autism-related phenotypes, because there's no way to say this is identical or not. So, for example, it has repetitive behavior, it has grooming itself to the skin off. And it has social interaction deficit, whatever that means in mouse. They have this defect. They also have sensory problems. Mostly we see sensory oversensitivity, so this is maybe different from human patient, but it's an abnormality. And you also have anxiety-like phenotypes and reduced locomotion, which you can say may be correlated with humans. And they also impaired learning. And motor learning is the most obvious. Other learning, cognitive learning, are very difficult to find. So we thought, oh, now we have all these phenotypes. We have the genes. We have the synapse defect. If we can find a mechanism why cause all this, then we have a target. Then we can develop. So we studied one by one, and so I'm summarizing it very quickly. First of all, what causes repetitive behavior? It turned out it probably at least involves the striatum, which is known very well. Also, striatum is for habit formation and repetitive behavior. If it's abnormal, we found it's in D2 medium spine neurons, one group of neuron. It's defective. Their synaptic function is reduced. If we increase that group of neuron activity, you can rescue that. But it does not rescue social. So that's kind of, okay, then what is the social defect come from? And one of my former postdocs went in, one now is a professor in China. They continue to work on this. They found that actually, at least the part of the reason is in the anterior cingulate cortex. The excited neuron in the cortex has reduced activity, so you can reproduce that or you can rescue that. And then, but that part only works with the social deficit, does not rescue the other parts. So for example, sensory oversensitivity. Sensory oversensitivity, it turned out is again, it's in the somatosensory cortex. Now this time the defect is mostly in the inhibitor neuron, because in this knockout mice, the inhibitor neuron activity in the sensory cortex is reduced. So you have hyperactivity in the sensory cortex, the pyramidal neuron. So these mice are actually, they use whiskers mostly, unlike us use eyes and hearing. And they are very sensitive. So as soon as the whisker touch something, they are startled. So now you have 3 different phenotypes, 3 different cell types, and 3 different brain regions. So how are you going to find one target that you can treat all of them? So it's kind of a job, it's really difficult. So, but it's not surprising because it's scaffolding protein, right? Morgan told us not 20 years ago, more than 20 years ago, it's a scaffolding protein. So why you're so dumb that you still have to do all these experiments? So it brings many different things at many cell types. So then what to do? How can we actually really develop therapeutics? So what do we think is that probably, at least in my lab and in probably other labs, we're thinking there are 2 approaches. For these monogenic disorders, it's one of the best ways, at least one of the best, is probably use gene therapy to correct the genetic mutation. Then everything else will fall into place. So it's probably the best. The question is, it's a neurodevelopment disorder. How early do you have to treat? Like if it's a development defect is permanent, can you reverse it? So the question is yes and no. So we did a mouse study that we give, because my study, we give perfectly genetic rescue, right? Because you have the genes turn on and off. So it's not like AAV, how many cells you can get in, which part are you getting, that gives you confusion. So we started with genetic rescue. We found that there are a few things you can rescue in adult, even mice, but most of them you cannot rescue in adult. However, the good news is postnatal, within first 3 weeks of the birth of mice, and it's hard to correlate with human, but at least during that period of time, you can almost rescue everything if you correct the genetic mutation. That means there is a postnatal window that will be very effective, at least in mouse model. So we moved that into, mice are mice. So we moved into a non-human primate. We spent literally more than 10 years to try to figure out a way to generate a monkey model until CRISPR came out and made life much easier. So we showed that indeed, I don't want to, I know Dan and Joe Nolan will talk about this, but I just want to show you one picture. So with the gene therapy, in the marmoset model, which is a non-human primate, whether 3 months or 9 months, 3 months is about 2 years old baby, and that's much easier to correlate with humans, and 9 months old marmoset is about 10 years old kid. So both groups actually can work really well, rescue both of behavior, including some of the cognitive functions. But most exciting to me is you can look at the resting state circuit activity. Do you actually change the circuit activity, normalize circuit activity? We all, as a neuroscientist, we know if you have a normal circuit activity, your behavior will be normal, most likely. So I just want to show this. If you can see On the top, these are functional, resting state function MRI, right? So we use subtraction to compare wild type with SHANK3 mutant. If so, red means that wild type is higher, it's reduced in SHANK3, and blue means higher in SHANK3. So what you can see is when you look at the wild type of SHANK3, you see the frontal cortical activity is reduced, which has been shown in mouse models also. But when you do gene therapy, correct also, it's almost like a wild type. So that kind of give us hope maybe will work in humans. So I don't have time to acknowledge everyone, but so there are many, many people involved, especially our team and many, many foundations, and I supported this work. Thank you.
SHANK3 gene function is very complicated, but gene therapy is direct and simple. And what a great segue to Dan Gallo.
Yep. Thank you. So first, just want to say thank you. Thanks, Caroline, for creating this excellent forum to bring us all together. This is, I think for us working on something like JAG201 day in and day out, the opportunity to be able to come out, engage with the community, engage with the academic collaborators is just incredibly meaningful. So big thank you. So I'm going to talk to you a little bit about Jaguar and the investigational treatment that we're developing to treat SHANK3 haploinsufficiency. In the interest of time, we'll just go ahead and move right into it. So Jaguar Gene Therapy was founded in 2019 with a mission to accelerate breakthroughs in gene therapy for patients living with severe genetic diseases. And the leadership team that brought this team together, much of that leadership team had previously built a company by the name of Avexis, which launched a gene therapy for spinal muscular atrophy by the name of Zolgensma. And so part of what they did was they brought over that expertise in manufacturing and CMC and had an early investment and focus on really differentiating the company from that perspective, with the goal of being able to target diseases that weren't just ultra rare, but potentially affected larger populations. And so that really opened the door to other diseases, including SHANK3 haploinsufficiency, type 1 galactosemia, and type 1 diabetes. Those are the three programs that are currently in development. You all know about JAG201, which is what we'll talk about here today, and I'll mention a little bit later. This was, we recently announced in January that we have IND clearance to begin our first in-human clinical study. But behind that, we also have JAG101 for type 1 galactosemia, and then JAG301 for type 1 diabetes, and we continue to work on both of those programs, and hopefully we'll be able to get to the point to move those into clinical development as well. So I know GUOPING just gave us an excellent overview on the biology of SHANK3 haploinsufficiency, but this is ultimately what we're looking to target with JAG201. JAG201 is an investigational gene therapy. It's meant to treat and address the underlying cause of SHANK3 haploinsufficiency, and so at its center, what we have here is the vector construct, but it contains a modified version of the SHANK3 gene. This is a mini SHANK3 gene, which contains, as you'll see on this next slide, all of the key binding domains. The intent here is to ensure that it can still recapitulate all the functions of SHANK3, but it's small enough at 2.1 kilobase pairs to fit into an AAV9 vector so that we can actually deliver it via gene therapy. And this was licensed from the Broad Institute at MIT and was developed by GUOPING. And so I want to give just a little bit of an overview of our preclinical development journey. And we're really excited to be at this point to be able to start to plan for dosing the first individual with SHANK3 haploinsufficiency. Of course, to get there, we have to make the case for JAG201 and the potential clinical benefit that it would provide. We do this by looking at this gene therapy in animal models, and we want to do a few things. We want to establish the efficacy, so we want to show that there's an actual benefit that we can correct in animal models. We want to make sure that we've got the right administration method identified, and so this is a part of our preclinical journey as well. And importantly, we want to understand what that safety profile looks like. right? And so the way we've done these studies, we've looked at a variety of models and types of animals. For preclinical efficacy, this was done in the model developed by Joe BUXBAUM, but it's a SHENG3 deletion knockout model. We chose this model because of the neurobehavioral outcomes, the motor coordination and motor activity outcomes, the sleep disturbances that you can identify in this animal model, which recapitulates known phenotypes of SHENG3 haploinsufficiency that we see in individuals. From the administration perspective, JAG201, our goal is to deliver this via intracerebroventricular administration, or ICV. When you do that, there's a few different options that you have in terms of determining what approach to take, and so we've done studies in non-human primates looking both at unilateral as well as bilateral administration. We won't get into these results today. These were previously published at ASGCT, but essentially what those results show is that you get similar levels of distribution of the gene therapy, whether you do it unilaterally or bilaterally. Unilaterally would be slightly less invasive, but importantly, similar levels of distribution, and because of the promoter that we have sitting just upstream of the gene, we also show in those studies that we get specific expression within neurons. And so while some gene therapy can make it out into the periphery, even though we're delivering it directly to the CNS, we don't see off-target expression in other tissues. From a safety perspective, of course, all of these opportunities to dose any animal with investigational gene therapy gives us important safety information. And so, again, all of this informs the safety profile. This is what we convey to the FDA, and this goes into the package of data that ultimately supports moving into clinical studies. But just to give you a sense, so those safety data consist of short and long-term data in knockout mouse models. Those also consist of dosing in non-human primates, which are unaffected, and so this is also beneficial because it gives us a sense of what are the potential safety outcomes or risks associated with overexpression in a non-human primate that currently expresses 2 wild-type copies. So what I will just highlight a little bit is we recently submitted our preclinical efficacy data package as an abstract to ASGCT. It has been accepted for presentation, so I can't present the specific data, but I'll give you a little bit of an overview, a little bit of a preview. We will present these data at ASGCT in a few weeks. But here we dosed SHENG3 knockout mice with a single ICV administration of JAG201 at different dose levels. So we want to look at outcomes. We want to show that there's a dose-dependent increase in the benefit. We also looked at mice for shorter periods of time, so looking at the functional behavioral changes over several weeks versus the long-term impact of JAG201 expression. Importantly, we want to show that any benefit that we see in neurobehavioral outcomes is sustained. And if we look out to 10 months, that we're also seeing durable expression, because again, with gene therapy, it's a one-time administration. We want to make sure that the patient's going to have a lifetime of benefit if this is successful. So in terms of results, we did show significant improvements in SHENG3 knockout mice following treatment in 3 key neurobehavioral outcomes. So we looked at a marker of sleep, of EEG biomarker of restorative sleep, so looking at delta power. We looked at motor coordination via the rotarod test. And then we also looked at motor activity and explorative behavior via the open field test. We show the deficits in the knockout model, as we would expect, and then we show corrective benefit after JAG201 administration. A few more things that we want to understand in these types of experiments. So are we expressing JAG201 at appropriate levels? And again, we see a dose-dependent increase in JAG201 expression within the brain. We do reach ultimately what approaches wild-type levels, as well as beyond that, depending on the dose that you look at. We demonstrate target engagement. So we've seen SHENG3 is a scaffold protein. It anchors, maintains many components that sit in the postsynaptic density. And we want to make sure that if we're increasing the expression of SHENG3 via the SHENG3 mini gene, that we're seeing increased recruitment of specific binding partners, and we show that there. Finally, we also show widespread distribution in these studies, and we've got 2 minutes, so I'm going to move a little bit faster. But more of those data will be available in just a few weeks at ASGCT, and we look forward to discussing them further. So earlier this year, January 31st, we did announce that the FDA granted IND clearance to move forward with our first in-human study of JAG201 in adults with SHENG3 haploinsufficiency. This first study is a phase 1/2 study. It's open label. It's a dose escalation. So open label meaning all individuals will receive the treatment. Dose escalation meaning we will test more than one dose of JAG201. The main focus here is to understand the safety and tolerability profile, but we've also, we're working to design this study to have robust opportunities to evaluate the clinical activity, the efficacy, potential of JAG201 treatment. We will deliver via ICV administration, and again, this initial study, IND clearance that's been granted, is in adults. And just a few additional details. I mean, this will be a small study, mainly because with the purpose of understanding safety, the learnings from this study will allow us to, more robustly define a potential pivotal trial. Because it is a small population, and as we've discussed this morning, there's lots of potential for heterogeneity in terms of the genetic background. We will have highly selective inclusion criteria, really to try our best to account for that heterogeneity. The idea being, if we're going to see a benefit, or if we're going to evaluate for a benefit, let's try to increase the similarity of these individuals so that we can have a better chance at understanding what that benefit might be. We've worked really closely with a lot of our collaborators. A lot of you are here. We're incredibly grateful for that. So the DSC, CureSHANK, PMSF have been integral partners from the beginning as we design our study, as we design our patient-facing materials, as we think about the outcomes, the data that we could potentially leverage from a regulatory perspective with the DSC, and we'll continue to do that. This is just a little bit of an overview of everything I just discussed. So, the first part of the study will really be 24 months. We'll, have robust opportunities to try to evaluate safety, try to evaluate early signs of clinical activity, and in line with the discussion earlier today, certainly, the goal is going to be to change the biology of the neuron, and so there's going to be potentially more appropriate ways to do that in the shorter term. If we're thinking about long-term changes in functional outcomes, functional, benefits, that's going to take more time, but we will continue to assess the potential outcome benefits in these individuals up to a period of about 5 years is at least what we're proposing now. And finally, I've mentioned the focus is adults. We recognize that there's an unmet need across this patient population, and there's been a lot of excellent literature that really helps us understand that the disease course, the disease manifestations begin early in life, and so the earlier we can intervene with a potentially efficacious treatment, the better. Our goal remains to be able to evaluate JAG201 in a expanded population, including pediatric patients. And so, thank you.
And so I'm going to ask a few questions or prompts, and the first is to James Fink. So PMS is a haploinsufficiency, as has been noted, thereby allowing different approaches to increase the expression of the remaining intact gene, such as ASO approaches. What do you think of that? And do you think we know enough about basic biology of SHANK3 and the gene structure to motivate such an approach?
Working? Yeah. So it's a great question. I would say there are sort of two buckets here. There's the question specific to an ASO for SHANK, and then there's the questions about ASOs in general. So let's maybe just focus on the questions around SHANK3 specifically. There are a lot of tools in the toolbox, as we heard today. So those tools have allowed for the testing of the gene therapies and the work presented by neurons. I think there's already a lot of great tools available for us. For ASOs, especially when you want to boost expression, there are certain mechanisms you want to target that are specific in some cases to humans. So I think this is a good point to highlight the human models. And Joe BUXBAUM made a great point earlier about the availability of models from different institutes and the need to sort of centralize the QC and the way that those things are reprogrammed over the course of the last 20 years. There's different ways to reprogram those. So I think there needs to be a focus on how we acquire and how we reprogram those cells to be able to unify the way that people work with these models. And that way we can use the human genetics to understand the SHENG3 gene in the human context rather than the mouse context. Because the molecules we're going to be developing are specific to human. And the ways we're going to modulate the SHENG3 gene in patients might not be the way that they are expressed in the mouse. And so although you could develop tool compounds to use in the mouse, it's really going to be the human models, I think, are the way to start to test the therapeutics for these patients. The other thing I think is important to highlight that was brought up earlier too is the expression of SHENG3 in the brain. So we show some nice fluorescent staining in mouse. There's talk about SHENG3 in the striatum. You know, striatum is a part of the brain that's sort of difficult for ASO to reach. So I think understanding what effects we can hit in the cortex. So if we look at cortex, we rescue SHENG3 in cortex, that would be important to do, and look at technologies that can enhance ASO penetration deeper into the brain. And then finally, maybe another thing to know is sort of how much rescue do you really need in, again, human context. Is it 10%? Do you need 50%? So I'd start there.
Next question is, oh, by the way, that was, I omitted to introduce, that was James Fink from Quiver. What a beautiful name for a company. The next person I'm going to call on is Yonghui Zhang from Yale. You're an expert on the structure of the SHENG3 gene, among other things. You have told me that mutating certain aspects of that gene might leave other parts of it intact. So what do you think about, how can you make some comments about that in the context of ASO therapy or gene therapy, replacement therapy? Are we studying the right genotype in mice? And are we going to fix things with the current approaches in the pipeline?
Thank you for the question. We kind of published, maybe almost 10 years ago, sort of know that SHENG transcript is very complex. And Dr. BUXBAUM showed one slide, and we realized that for sure at that time we know that's incomplete due to the technique limitation, because SHENG3 is pretty long. You can have 8 to 10 kb transcript. So the short read sequence we really now cannot assemble the whole transcript until a couple years ago. I think some of you probably saw the poster presented by Xiaona Lu and the research associate in the lab, and get a sense how complex it is. And with her talent a couple years ago, so she really did an elegant work where you can capture the SHENG3 transcript. Also, we capture the SHENG 1, 2, 3 all together. Then she did a long read sequence, used PacBio. So now we think we have a pretty good understanding the complexity of the structure, I think supporting GUOPING's early conclusion. That's very complex, more complex than we thought initially. So now we say, okay, that's a several hundred isoform. Then a few things we learned. One is, it is complex. Second, there's additional exon which fuse two genes together adjacent. That's unexpected finding, which means SHENG3 has more than, typically we say 22 exon. We say now maybe have a 30 exon for some isoform. And the last is for the, our data may suggesting some of the animal model may not have a molecular validity to test the therapy. So that's probably a good indication for when you're testing the therapy, which model you're going to use. The same thing when you use antisense oligo, which isoform you're going to upregulate. The challenge is also prove Dr. BUXBAUM's point, like we get pretty complex. Do we really need to know every detail of this complexity for the human translation? That's probably not. But we just need to figure out what's the key component that may affect translation for this complexity. Understand it better, I don't have any answer for that for sure at this point, which tells you that's complex, that's a structure, more complex than you thought. So just go back and look in your individual model in the study and see if you can reinterpret that data or do more work to prove your point. And the same thing I was looking for earlier for the first slide presentation by Casey from the Sinai about this human phenotype. So it was surprising to me, she validated that point, which is small deletion permutation for the psychiatric 2 behavior domain more severe than the larger deletion. So I was sitting there saying, okay, what's the interpretation for that? So I have my view, and then some of you have your view, and you look at the mouse data, you disrupt one isoform, you upregulate other isoform. So that's what we observe in some of the mouse model. So the question is a truly phenotype, not truly loss function in the mouse model you analyzed. It's a combination of loss function plus the gain of function for some isoform, normally not expressed that high. So that creates a lot of challenges for the interpretation for a lot of, we collectively, GUOPING, me and Joe and many other create amazingly a panel of SHENG mice and other mutants. It's probably the only first disease model. You have more than 16, 18 different line of SHENG mice. It's a real careful choosing which one you do and then kind of get a sense for that stuff. Yeah.
So the next question that I was fed was meant for Joe BUXBAUM, but I noticed he avoided coming up to the front.
Joe,
but that's fine, that's fine. Yeah, so, you know, maybe other people can also chime in on this. It's a very frequently asked question in the translational neuroscience field, which is, how good is the phenotype overlap between, you know, mouse models and human patients? That is such an open-ended question that maybe I can rephrase it as, what phenotype do you think is found in a mouse that is most relevant and clinically relatable to the patient suffering?
I would say, unfortunately, the phenotype in the mice is not particularly striking. Spend any amount of time with families, and you'll know there's a huge gap between what we see in the mice and rats so far and anything we see in patients. That's one of the reasons that people are focusing more on the homozygous knockout, where both alleles are disrupted. And obviously that creates some kind of issues about construct validity. We can't avoid that, but the reason people are doing that is because the phenotypes in the mice are minimal. Certainly, the bigger the deletion, we think comparing at the same time, the small deletion, big deletion, there are more phenotypes in the mice. They tend to be more in the motor domain. And actually in electrophysiology, the phenotypes are quite strong, LTP, LTD, things like that. But, you know, there's not much of a phenotype, and this is not unique to Phelan-McDermid syndrome. The Fragile X mice for decades didn't have much, and they've had to find some subtle changes that kind of became the endpoint for Fragile X. But remember, in Fragile X, one of the things some of the habituation studies in the mice were seen in humans, in mice, and in slices from the mice. And so even electrophysiological deficits or motor deficits or small deficits can be useful for validating an approach. And now we'll hear who disagrees and agrees. Here next. Oh,
maybe I can ask GUOPING. If I remember correctly, the non-human primate stuff is done in heterozygous mutants, correct?
Correct. Yeah.
Right.
Both macaque and marmoset, we only use heterozygotes, mimic human conditions. They all have phenotype. That's a very big difference between the mouse model and the monkey model.
Can you say one or two, you know, sentences about the most striking phenotypes of the heterozygous mutant monkeys?
Yeah. So in the marmoset, we have fully characterized. There are quite a few things from many different domains. For example, they are very. If there's you can put a marmoset in their home cage, then you introduce another marmoset, but they cannot touch it. It's a small cage. They will be very afraid of any new things, strange things. Like, they like touch screens. In the home cage, they have an iPad. They like go touch different, things, then get a reward. They're like marshmallows, so we get really teeny tiny marshmallows. And if you put any kind of shape or square, they will go touch it. But if let's say you change it to a marmoset face, they will still go touch it, but they prefer stay way back and occasionally touch it. So then I think the most important is their anxiety level. Anything new, introduce another marmoset, anything, they will basically startle and go back and just avoid all these things. And they also don't understand the social situation where marmoset calls a lot. They're very noisy. They talk a lot all the time. When I saw marmoset, I went in front. They all come look at me. Then they all went back, talk about it. Maybe they're talking, who is this guy? Why is he? And then they come back and look at you again. So they talk a lot. They have reasons why talk, like social calls. The call usually is a marmoset leaves. Then you couldn't see it. Then they will call. And all these are very structured, but the SHENG 3 heterozygous, they don't understand that. So when you have another marmoset next to you, why do you need to call? You can just social by grooming or other things. They will startle and call, so make wrong calls. So all these things are quite obvious in that sense. But there's a range, always like humans. Why normal marmoset has a range of behavior, then mutant has a range of behavior. What we found is the best way, because their behavior is affected by cage weight, by noise in the room, because if it's a very jumpy marmoset, a normal marmoset will affect another normal marmoset. So the most robust thing is compared to their cage mate, and how much different they are from the cage mate. So they are significant phenotypes in heterozygous.
Right, and is that reversible by gene therapy in the marmoset?
Yeah, so most of them can be reversed. And they also have a very significant learning disability, which we couldn't find, hard to find even in homozygous mice. We published, said we didn't find it learning in the homozygous mice. So you basically give a touchpad. They know they need to touch. There are two phenomena very obvious. One is they have restricted interest. They like to touch just one corner. The rest they don't like to touch. Then we say, okay, that's a difficult. Then they become randomly accurate, not they are thinking. So we force them to touch the rest. If you keep touching the same place, you don't get a reward. So they gradually touch other places. So they learn much slower. They make a lot more mistakes. Then you can use a heat map, look on the iPad. What did they do wrong? They know actually they will touch it, but they don't really understand you have to touch the middle part of that. So they touch it outside. And gene therapy, you can see by heat map, they all move to the middle. Now they understand they touch it. So, but there are things that we could not rescue. Most obviously is a very complex cognitive function testing, for example, you based on the intensity of the flashing, then you can decide what time you need to touch it to get reward. And so there are things you cannot rescue, which we think is much more difficult cognitive learning. And we don't know is because we did not get enough cell or we did not get it early enough. So there are things, it's not like, oh, it's because of mini-gene, like Yonghui said, maybe you need everything to make it perfect. So all these things, actually, there are some defects we could not, but it's on the high end of cognitive function aspect. Other motor, everything, behavior, at least in the marmoset model, we can rescue them very well. So.
Thank you. Well, it is now noon, but I just want to ask maybe one last question, and then the audience can attack these panelists in the lunch. So the last question is more for the company folks. So, and it's about biomarkers, and I'm not talking about efficacy or surrogate efficacy biomarkers, but more about pharmacodynamic biomarkers. Especially when you have a trial that is open label, and that has a high chance of non-success, shall we say. How do you know that your drug has hit the target? Are there any molecular biomarkers that are more proximal to the target than EEG that tells you that with your gene therapy, you've actually increased SHANK3 protein expression in the brain?
So I'm not aware of any, biochemical biomarkers. At least Jaguar's not evaluating any biochemical biomarkers that might be early indicators of change in neuron biology. I think at the moment, our focus is on, what we understand from the small studies that have been done, what Alex shared earlier today, right? Trying to understand at an EEG level, are we changing the biology? Are we changing the ability of those neurons to interact, to communicate? That's really where our focus is at the moment.
Well, I guess that's the answer I expected, but I'm coming from a industry background where we want to know if the trial fails, whether the drug failed or whether you just didn't do what the drug was meant to do. That's basically my point. If you don't get an EEG movement and you don't get clinical change, maybe your gene therapy just didn't work, is what I'm saying. How do you try to de-risk that? Are there any, maybe I can open this up because I know it's an unfair question, Dan. Are there any preclinical model studies that could be used to discover some molecular biomarker that says, I think I have increased SHENG3 expression by 30% in the brain with this gene therapy?
Maybe guys comment on the other trial, as probably the Angelman trial that much advanced for a certain degree for their ASO. That's a real challenging one, but they try very, very hard, particularly in human, try to search for the CSF marker for reactivation, but after many years, many $1 million investment, and so far has not been very successful for all different kind of reasons for the gene expression profile. But I just don't think we have done any sort of SHENG3, maybe GUOPING other. So it's just, you can detect SHENG3 in the CSF or not. To me, it's the first question. If you have a baseline CSF, you can detect SHENG3, and that may be the first thing you can easily to look into and to see if a gene, particular gene therapy or gene correction, that whether your correction is actually reflecting that. I mean, you cannot go to the brain unless you, sometime right now we looking for all this high sensitive PET imaging marker possibility. So, but it's a very, very good question. All this brain-related disorder, it's all come down to that question. Behavior is not truly very, very robust or reliable, and this particular, if FDA asks you everything to the older age first, before you move to the 4 years age, the 2 years age, are you gonna, besides to prove the safety, and you're not gonna see much benefit by 20 or 40 years old first trial for that purpose.
So I want to add, we actually collected the wild type and the SHENG3 CSF monkeys, and it's easier to do longitudinal with macaque monkeys. So actually, it's in the freezer, waiting for mass spec analysis. So we don't have results yet, but it has been in the freezer for a while. So we did collect them, and is it possible to do that kind of thing?
Yeah, no, that could be a good resource for discovering something that could be shifted with gene therapy, right? Okay, time is up, and Geraldine is approaching the podium.
Time is up. Thank you to the panelists. This is great. I feel like we needed 2 days for this panel, and we needed 2 days for the earlier panel this morning. We thank y'all for doing the impossible and describing all of this work to our audience. We're going to break for lunch now. We're going to reconvene here. If you would, try to be back here at about 12:40, and at 12:45, we're going to do our first ever awards ceremony. So we're looking forward to that. So go and enjoy your lunch, and we'll see you in a few minutes.
Hi, everyone. I'm Kate Still. I'm the Scientific Director of the Phelan-McDermid Syndrome Foundation. Thank you so much to CureSHANK for inviting me to present. I apologize for the hotel background. I am traveling as I record this, And I so wish I could be there in person with you all. I know how much important work is ongoing in drug development, and I wish I could be there to discuss it. But I will be presenting virtually some of the current data sets that we have at PMSF and how they might assist you in some background information on the population for your efforts or leading up to clinical studies or even during them. So we have a couple of different data sets and they serve different purposes. So I hope to be able to clarify that a little bit. I'll share screen. So for the numbers of people with Phelan-McDermid syndrome and where they are in the world, the best place to find that is our membership data. So the membership at PMSF is actually the largest count of people with Phelan-McDermid syndrome that exists worldwide. We are currently at approximately 3,600 families and counting. And that number represents, each number of that a distinct, family or person with Phelan-McDermid syndrome. So it's not further diluted or decreased in any way. We are gaining more than one new family in our membership per day, which is really exciting. And that rate has increased in recent years. And we ask a lot of demographic questions, location, age, gender, and we're pretty frequently updating this information, cleaning that data. So if you would like the most current snapshot, please reach out to us at info@pmsf.org or directly to me at kate@pmsf.org, and we can provide that. One note is that membership does not require a genetic report to enroll. We do collect genetic reports as part of our registry and our IRB protocol, so I will discuss that in a second, but membership does not require it, so we cannot guarantee that every member has a confirmed genetic diagnosis of Phelan-McDermid syndrome, although we are aware that the overwhelming majority do. If you would like some more information on genetics, the best place to find that right now is the PMSIR, or Phelan-McDermid Syndrome International Registry. This is a data collection effort that is owned and organized by the PMSF. It ran from 2011 until 2020 when new data collection ended in order to improve the registry. Families uploaded genetic reports onto an online platform, and these reports were then curated by genetic counselors and genetic experts into the relevant information. And we now have an export of over 500 people with Phelan-McDermid syndrome and genetic information that we can share. It requires a painless application of just background on the study team and some of the outcomes that the study team is looking to achieve, and signing a data sharing agreement. The PMSIR also collected demographics and medical survey data. These have been very helpful in having great outcomes, and a couple of publications have been published or are being published even more recently that may be presented at this meeting from the PMSIR data. But we also heard from scientists that the data was sometimes difficult to analyze because not all questions were required to be answered, or questions came from different surveys, so they weren't entirely standardized. So we have been working to improve the registry since getting that feedback. And that version of the registry that is an improvement is now called the PMS Datahub. It can be thought of as a continuation of the PMSIR because we are retaining the information from the PMSIR, and it's the same IRB protocol that's been improved, but we are collecting new information, have made some changes. So we're on a new platform. We are also using standardized surveys such as ClinGen Genome Connect surveys, including a general health survey. The reason for that is that's used widely by other groups. For instance, if you've heard of RareX, an online data platform for a lot of rare disorders, that is one of the main surveys that they use. And the survey asks some general body system questions such as, does the person with Phelan-McDermid syndrome have issues with their eyes or vision, or their nervous system, or their endocrine system, etc. If the answer is yes, the survey expands into more detailed questions on specific diagnoses in that area, as well as date of or age of diagnosis and things like that. So we have data from that survey on more than 200 people at this point and hope to get more people enrolled. And we also have the ClinGen Genome Connect genetics survey, which is currently being curated by genetic counselors also. And this includes both people who have just enrolled in the Datahub anew, or the reports from the PMSIR. So that's a pretty significant number of reports. And the genetics survey is also very detailed. It includes a lot of questions about structural variants and sequence variants seen in Phelan-McDermid syndrome, and some summary data there as well that classifies these genetics into general classes. We are also collecting information on medications and demographics. So although demographics is being collected across all three of these data collections, the membership can be thought of as the largest number, and these data sets from the registry can be thought of as smaller numbers of people, but more of a deep dive into this medical and clinical data. And we are also considering adding new surveys at any point to the Datahub. It does have the capability to do this. We want to be very careful in choosing these so that we don't add a lot of time and survey fatigue for families, but we would love to talk more with our scientific and medical partners to see if there are certain outcome measures that may be, very long-term, we know they're good, that we would like to include in the Datahub. We can do that. This data from the Datahub itself is not yet ready at this present moment for request. It is definitely getting there, and we have come a very long way. So some of the reasons that it's not ready quite yet is that we're still getting through the backlog of PMSIR reports from the genetic counselors. They have done a majority of these, and they are looking to complete that work over the next couple of months. And we are also looking to clean the data and have a very high quality data set before sharing it with researchers, which includes a robust data dictionary and deduping and cleaning of that data. So that is what we're working on now. And I think that this data should be ready within the next few months. And please check in with us if you would like more information on the Datahub and what it can give you before then. And then I will also update when that data is ready. But in the meantime, please contact us if you need any information, info@pmsf.org, or to me at kate@pmsf.org. We are also working to increase our biosample collection at our upcoming conference this July. We've been chatting with Mount Sinai, who I know is also attending this meeting, in hopes of collecting samples and having iPSCs and iPSC-derived lines made long-term to really bolster that data set, which I know is really important for you all for drug development. And if you are interested in our conference, please check out pmsf.org/conference. And thank you all for your time. I appreciate it.